Hello learners, I'm Dr. Poonam Agrawal, Professor Biochemistry. In this video, I'll be discussing regarding the difference between synthase and synthetase enzyme. The first line is they are not synonym. They are different enzymes belonging to different class, having different role to play, and their requirement is also different. So let's talk about it. So for that, I will explain you in this manner. This is synthase enzyme and this is synthetase enzyme. First of all, you should know that synthase enzyme belongs to class 4 of enzyme classification. What is class 4 of enzyme classification? That is lyase enzyme. So synthase is a lyase enzyme and hence the enzyme commission number which is given to it is 4 because it is belonging to class 4. On the other hand, synthetase is the enzyme which is the member of class 6 enzyme of enzyme classification, that is ligase enzyme, ligase class of enzyme. It belongs to ligase class of enzyme and this is 6th class of enzyme, so enzyme commission number is 6 given to it. When you're talking about lyase enzyme, what it does, it breaks a compound in its component, which may be a reversible process or may be a irreversible process. If the direction of reaction is towards the cleavage, you name this enzyme as lyase. And suppose if the reverse reaction is followed, because it's a reversible reaction where two compounds are condensing to form you the another compound, you can call it as synthase enzyme. You can nomenclate this enzyme as synthase enzyme. So synthase enzyme is condensing two compounds together without the need of any source of energy. You write down that no need of energy is there for condensation by synthase enzyme. No need of energy. Generally, nucleotides provide the energy like ATP, GTP. Such things are not required here. When we talk about synthetase enzyme, this particular enzyme, it belongs to class 6 ligase. And what the members of ligase class of enzyme they do, they ligate two things together with the help of energy which may come from nucleotide. So number one, they need nucleotide to provide the source of energy. It may be ATP, it may be GTP, etc. So synthetase are ligases, and ligases are the enzymes which unite two things together with the help of some energy which is coming from nucleotides. So source of energy is required. Now I would like to go on important examples of these enzymes. Synthase. The enzyme aldolase, the enzyme fumarase, the enzyme arginosuccinate, lyase. Then PDH complex enzyme, glycine synthase enzyme, fatty acid synthase enzyme. They all are lyases or they all are synthases. And if I talked about example of synthetase, the glutamine synthetase, arginosuccinate synthetase, carbamoyl phosphate synthetase, 1 and carbamoyl phosphate synthetase 2. They all are synthetases. Acyl CoA synthetase, which you otherwise call it as thiokinase, DNA ligase is class 6 enzyme. This also falls under this category. Malonyl CoA synthetase, which otherwise is called acyl CoA carboxylase, which is rate limiting enzyme of fatty acid synthesis. It also belongs to this category of synthetase. In fact, all carboxylases, they belong to class 6 ligase, and they all are actually synthetases because carbon dioxide is added with the help of external source of energy. Nucleotide is needed. So this in nutshell is the difference between synthase and synthetase. So you should clearly know that they are not synonym, they belong to different class, and synthase, they do not need energy. That is most important statement about them, and synthetase, they need external source of energy. So all the enzymes which are belonging to class 4, if they are condensing two things together, because ligases are reversible, so if they are condensing two things together without the need of external energy, they will be called as synthases. And aldolase, which we see in glycolysis and fructose catabolism, fumarase, which we see in TCA cycle, arginosuccinate lyase, urea cycle, PDH complex and oxidation of pyruvate, glycine synthase, fatty acid synthase, they all are belonging to this category. And when I talked about synthetase, the glutamine synthetase, which is needed to attach the ammonia on the glutamate with the external source of energy, arginosuccinate synthetase in urea cycle, carbamoyl phosphate synthetase 1 in urea cycle, CPS2 in pyrimidine de novo biosynthesis, acyl CoA synthetase or thiokinase in activation of fatty acid for carnitine shuttle, then ligase enzyme during DNA replication and repair, malonyl CoA synthetase enzyme during fatty acid synthesis. They all are this is how are classified as synthase and synthetase. Thank you very much, and for detail on individual statements you can see other videos on my channel. Thank you very much.